Welcome to Glioblastoma, aka GBM, a podcast brought to you by the Glioblastoma Research Organization, highlighting stories of GBM warriors, caregivers, medical advisors, and more. Join us this season as we connect with members of our incredible community and have meaningful and insightful chats regarding all things glioblastoma. Please note that any information provided on the show is not meant to treat, diagnose, or prevent any disease, and all information that is discussed in our conversation is what worked for the individual themselves and should not be taken as advice. The information provided on this show is not a substitute for professional medical advice, and you should contact your medical provider and healthcare team with any questions. Dr. David Ashley, welcome to Glioblastoma, aka GBM.
Pleasure to be here,
It's
and
great
thank you
to
for
have
inviting
you.
me.
Absolutely. So you are the director of the Preston Robert Tisch Brain Tumor Center, but I was told in a past life you were also a professional surfer. Can you talk to us about that?
Oh gosh, come on, give me a break. Really?
Yeah.
That's where you want to start? Well, no, I misspent my youth, spent a lot of time in the water, and did a bit of surfing, but actually was persuaded by my father that it was never going to hold a future for me, and actually I was a better academic than I was a sports person. So he persuaded me to go to university.
So was that the transition you had into neuroscience?
No. So when I went to medical school, we went straight in as an undergrad. In Australia, we went straight from high school into university, and we did 6 years of medicine, and that's a generic undifferentiated medical degree. And then from there, I did internal medicine and so on. And sadly, when I was in my final year of medicine and my intern year, my father became very unwell and actually ultimately passed away from a terrible cancer. And it was really in the second year of my career that I started becoming obsessed by cancer. And so I trained in cancer, and initially I trained in pediatrics, but ultimately it was clear that there was a huge need in pediatric brain tumors at the time. I was working in Melbourne, and only 10% of children at that time were surviving, whereas the kids' leukemia was, much more curable. And so I was fortunate enough to receive an Australian government scholarship or fellowship that allowed me to train anywhere in the world. And I came over to the United States and decided to train in brain tumours. And I don't know if you know, but from 93 to 97, I spent my time doing postdoctoral research. I'd done a PhD in Australia as well with Daryl Bigner, who was my predecessor.
And that's how you got to Duke.
There you go.
Okay.
That
Got
was
it.
Yeah. And then I ultimately finished I worked as an attending with Henry Friedman. And then in 97, went back to Australia and worked in Australia for 20 years. But my journey to neurosciences was really a needs-based assessment of the field and an opportunity around, that things were going really badly in brain tumours at that time for people. And also, probably feeling the personal or reacting to the personal grief of losing my father to the cancer.
You mentioned having done your initial training in pediatrics, but now you have focused more on adults. What sparked that transition?
Yeah, I often reflect on that. There's two extremes clinically that I really enjoy working with. I really enjoy pediatrics because of the family-based care and talking to people, but I also enjoy looking after older adults, funny enough. And so I started working in pediatric brain tumours, but also realised that there was a critical mass issue, and that, if you really wanted to make progress in the science, we could, at the time, we thought we could do things more quickly in adult glioblastoma because we had more patients. And I really enjoyed looking after elderly patients at the time. So I moved from pediatrics and I did some formal training in adult neuro-oncology, and ultimately got credentialed in Australia, grandfathered in, if you will, as a medical oncologist. So I had a dual certification in pediatric oncology and medical oncology.
Okay. Another question I had was, you're a professor of neuroscience and neurosurgery and oncology. How do you have all these different professions, and what did you decide to, how do you choose like what to focus on?
No, they're academic appointments.
Okay.
So I'm trained as a medical physician in medical oncology, but I'm appointed in the department of neurosurgery, and I have a professorship in neurosurgery because of the academic side. So that's really based upon my research. I'm not a neurosurgeon, right? And in fact, I think it would probably be a dangerous thing if I was a neurosurgeon.
It's funny.
I'm too impatient.
Henry said the same thing.
Oh, did he? Yeah. I'm too impatient.
How do you feel working alongside Henry, being that you partnered together so many years ago? How has that transition been from now, like having been partners in the past, and now you're the director of the Brain Tumor Center?
Well, he was my boss.
And now you're his boss.
Yeah, well,
roles reverse.
I don't think anybody can ever be Henry's boss, right? But I love Henry. I think he's such a unique character, and he has made a mark in the world that is indelible. I find myself fortunate to be both a friend and a colleague. I think there's brilliance to Henry that in some ways people might miss, but I love it, and he doesn't have a big ego when it comes to that sort of hierarchical structure. He doesn't feel a need to be in charge of everybody. I think he's comfortable enough in his own skin that we work well together.
Mm-hmm. That's amazing.
Yeah.
When you got to Duke, originally going back, your focus was on in vivo and in vitro studies. Mm-hmm. For those that don't know the difference, can you explain what that is?
So in vivo is in life. So that generally in the lab, that means we're doing animal studies where we try and recapitulate what's happening in human brain tumors in animal models. And in vitro means in the dish. And so this is research that's done at the bench side in general. So we do both. And each of those different things has strengths and weaknesses when we're trying to model and understand human diseases.
When starting a research project, will you always start in vitro and see how that progresses and then move to an in vivo study?
Generally, I think that's true. But also, we look at the models and we decide what is going to be most informative, in particular to the question that we're asking. So if it's a question of biochemistry, or if it's a question of understanding how something works on a particular cell, the simplest thing may be just to do it in the dish. But if it's a question where we're asking, well, how does the blood penetrate the blood-brain barrier, or how does the drug or the therapeutic distribute through the body, these are studies that can only be done in vivo, in animal models, and then ultimately in humans.
Mm-hmm. How does a therapy or project translate from the animal model to humans? Like, at what point do you guys decide as researchers that, okay, we're taking this to the next level, and then how does that work?
Yeah, I mean, there's no particular fixed benchmark, and each of those decisions depends upon an accumulation or a body of evidence. So usually what we try and do is, if we're moving from in vitro, in vivo, and then into people, we'll try and get at least 3 or 4 different angles at the same question so that we can have a high level of confidence that it will translate, and particularly it won't hurt people. Mm-hmm. So we might use 2 or 3 different animal models to ask the same question, so that it's not just a particular peculiarity of one animal model, but it seems to recapitulate, keep using that word, over and over again in multiple models. We also, there's what we call efficacy questions, where we want to see that the drug or the intervention's actually working in the animal model, and we'll do 2 or 3 of those. And then we might do some what's called toxicology, where we give the drug to mice, and we may give it to other animals as well to see if the drug's going to have a toxicity that's unanticipated. And so they're the sort of the precursors of getting to a level where we can get approval to do human studies, and that's usually through regulatory agencies like the Federal Drug Authority.
Why is it that mice are the main subject?
Yeah,
those models?
This is becoming a really big question. Okay, it's a really great question. I think as time goes by, it's going to turn out that some of the decisions we made with, we call it murine or mice studies, have really been ill-advised. And what I mean by that is that mice don't model well human brain tumours. And often the mice studies are overinterpreted. Why mice? They're cheap. They have litters of pups of manyfold. You can breed them. You can do genetic engineering at a relatively low cost in a mouse. And so you can get a lot of information for a relatively low cost base. That's one of the main drivers. There's also a massive resource of different mice with different genetic defects and so forth that you can purchase, interrogate. And so it's utilitarian in some ways. The problem is that in brain tumours in particular, we're learning that the mouse brain tumour does not really reflect well the immune system of a human brain tumour or the way a human brain tumour reacts to therapy. So I think we've been tricked over decades with some of the outcomes in mouse studies. I think more and more we're understanding that we need to go more quickly with, but prudently into humans, because ultimately you have to do these studies in people.
Yeah. That's so interesting. If there was I guess it's also an ethical question because,
it is.
You can, there's so many different animals out there, but I guess choosing to, do this on an animal, I guess it's challenging.
We've been doing models, there's some work that we're doing where we're looking at what's the seed cell of the, and other groups are doing this as well, where we're looking at, what's the original cell that ultimately forms a glioblastoma. And I think what our modelling is pointing to is it comes from a region in the brain called the outer subventricular zone.
Where is that?
So the ventricles are the draining system of the brain, and then there's a region that's called the subventricular zone, which is just outside the sort of fluid area. It's the very thin layer that sits just outside the ventricles. And the outer subventricular zone is just outside that. So mice don't have an outer subventricular zone. If you look at a mouse brain, actually it has a very smooth surface, whereas if you look at a human brain, it's quite convoluted. It almost looks like worms. Like it's and that's because the human cortex is very, very complicated, and the outer subventricular zone accounts for that complexity. So mice don't have a complex brain because they don't need a complex brain. Humans, we need a complex brain. That's how we've evolved. And so, one of the thoughts is that the cost of evolving a complex brain is glioblastoma. And that's why mice may not reflect the human brain.
But wouldn't you say that there has been significant research and a lot of really great studies. So some that have come from mouse trials, would you say that's more of if there's, is that luck? Because if their brains don't model it, how is it say that project didn't work?
No, it's a great question. Has there been great studies? Let me reflect back to you. What progress have we made in glioblastoma in the last 3 decades?
Well, according to my PhD.
Yeah.
Well, is it Temidar? That was in the last 3 decades, right?
Sure.
Yeah.
So you can take, it was, Avastin was as well. The drugs that will work, most chemotherapeutics will have an effect in a mouse brain tumor model, and Temidar certainly did. Mouse brain tumor models are really good for modeling, for example, blood-brain barrier. So if you want to choose a chemotherapy that will pass the blood-brain barrier and have an effect, Temidar was a great choice because it passes the blood-brain barrier, that modelled in a mouse, response in a mouse, and then ultimately a response in the human. But the drug itself was originally made, I think, probably in the 1970s. I'd have to confirm that. And it's a monofunctional alkylator based upon a drug called DITC, which was developed in the 1950s. It wasn't specifically made for brain tumours, but somebody took it, they put it. So, okay, I'll give you that one, tick to amber, right? But if you look at the models, for example, of immunotherapy in mouse brain tumours, they grossly overestimate the efficacy of immunotherapy. A lot of the mice studies have shown that checkpoint inhibitors work, that other things work. In my own studies, we've modelled vaccines and other therapeutics in the immune system, and they work beautifully in mouse systems, but none of them have translated when we get to the human system.
Really? Biodexa Pharmaceuticals is proud to sponsor the Glioblastoma, aka GBM podcast. Biodexa Pharmaceuticals is a small biotechnology company hoping to make a big difference in the treatment of glioblastoma. Using their proprietary nanotechnology, Biodexa is developing liquid formulations of an investigational drug which can be delivered directly and locally into the tumor via an implanted catheter. This drug has been previously investigated in pediatric patients with brain tumors. Biodexa Pharmaceuticals is currently running a clinical trial in patients with recurrent glioblastoma known as the MAGIC G1 trial. To find out more about the MAGIC trial, visit magictrials.com. Imagine waking up from brain tumor removal surgery knowing that your radiation treatment is already underway. That's how gamma-tile therapy works. At the end of brain tumor removal surgery, the neurosurgeon implants the gamma-tiles where the tumor is most likely to return. So instead of waiting to start daily standard radiation treatments that go for weeks, you get a head start against tumor cells and get back to your life sooner. For operable brain tumors of all types, including glioblastomas, brain metastases, meningiomas, gamma-tile therapy is a one-time, targeted radiation treatment with fewer side effects and far less chance of hair loss than external radiation. Gamma-tile therapy is tough on tumors and easier on patients and caregivers. Learn more at gammatile.com. Gamma-tile therapy is an FDA-cleared radiation therapy for patients with newly diagnosed malignant brain tumors and recurrent brain tumors. Novocure is pleased to support the Glioblastoma, aka GBM podcast. Novocure strives to extend survival in some of the most aggressive forms of cancer through the development and commercialization of their innovative therapy called Tumor Treating Fields. Novocure partners with the Glioblastoma Research Organization to work together on behalf of patients and their loved ones impacted by GBM. To learn more, visit novocure.com. Rune was built by a team of patients, caregivers, and medical experts, consisting of neurosurgeons, neuro-oncologists, psycho-oncologists, radiation oncologists, nurse practitioners, and social workers who have devoted their lives to treating and helping glioblastoma patients. For anyone navigating GBM, Rune offers a wealth of medically vetted, digestible video answers to common questions. Answers are organized into major topics ranging from surgery to radiation to caregiver mental health. Check it out at rune.com.
What do you think is needed in order to make these therapies, I guess, translate better to humans? Is that just more quicker translation from in vitro to in vivo, but human wise?
Yeah, I mean that in part. But the other thing is, I think we may be able to get to better animal models as well. So, for example, pigs have an outer subventricular zone. Dogs have an outer subventricular zone. There's ethical issues, and large animal studies are much more expensive than murine studies. But or we may be able to develop mice that have the more appropriate cell of origin, and then model from there. The immune system in a mouse brain is very different to the immune system in a human brain. So we need to come closer to what's going on in a human brain.
And talking about, vaccines, this different immunotherapy, the polio trial at Duke is coming back. So Henry told me about, and there were some questions that he was not able to answer. So he wanted me to ask you. The first one was regarding where it's placed in the body, how it's going to be injected. The chemo is on the same side lymph nodes versus just anywhere, since the lymphatic system goes everywhere.
Yeah.
Why is it being that it's on the same side?
Yeah. So this is work that Mike Brown and Matthias Gromeyer have been doing, and other people as well in other institutions. What has been discovered in the last decade is that the brain has actually a lymphatic structural immune system, which is very similar to the rest of the body. There are some differences, but that the primary place that the immune system of the brain drains into is the cervical lymph nodes. And the apical cervical lymph node in the cervical chain.
What is apical?
It's the top of the lymph node chain in the cervical system, is where it's the first lymph node that either T cells or antigen presenting cells trafficking to and from the brain enter. When Matthias and Mike have injected polio into the brain tumour itself, it seems that a multi-dose format is much more effective than a single dose. The first study of polio that we published, we just did single dose, and there did seem to be some efficacy for some patients in that study. In the animal models, it seems that multi-dose format is much more effective. And in fact, there's been a Japanese study of herpes virus now that also seems to indicate that multi-dose oncolytic viruses are more effective than single dose. And that's a human study. That's in people.
Wouldn't you be concerned though? Like, again, I'm not a doctor, so I'm not sure like terminology of this, but like if you're, injecting herpes into someone, like maybe you're, helping a brain tumor, but wouldn't that give you other parts of another virus? Like if your immune system, like let's say I can't fight all of that.
But these viruses are all modified, so the brain damaging or the tissue damaging parts have been engineered out.
Oh, okay, got it.
So the polio virus that we're using is a modified polio virus. It's called PVS Ripo, and the nasty parts have been engineered out by Matthias and have been replaced with the simple cold virus.
What is that?
Rhinovirus, the thing that gives you a runny nose.
Okay.
Yeah. So yes, you are injecting a live virus, and there is some risks in that you need to balance. In the case of polio, of course, polio is a devastating neurologic disease. But what Matthias did was he engineered out the parts that cause the bad stuff and left in the parts that inflame the immune system. And so it's a benign virus. So, I'm presuming you and I, we've been vaccinated against multiple different things. They're in general, they're modified viruses that have had the bad bits destroyed or taken out.
Got it.
Yeah. But back to your question, just to tidy that up, multi-dose platform seems to work better than single dose, but it's pretty difficult to give someone 6 or 10 injections of polio into the brain. So what Matthias and Mike have discovered is actually it's just as effective to give the polio virus into the cervical lymph node, and it creates the same as a multi-dose platform into the brain. Why do it on the same side? Seems to make sense.
Okay.
Right. I mean, we think that the right side of the brain drains down to the right lymph node chain. I mean, it could be that you could do it in the left as well.
Because I know, like, if you have like a tumor on the right side, it affects the left side of your body, and vice versa.
Oh, okay. That's but that's because the motor neurons cross over.
Got it.
When they run down into the spinal cord, whereas the lymph nodes drain straight down.
Okay.
So it's
learning so much. This is great.
Well, surprisingly, this has only been learnt in the last 5 to 10 years.
Okay.
Yeah.
When is that trial opening up again?
I think it should open in the second half of this year.
Oh, that's amazing.
It's very close.
And are there other sites, or is it just a Duke project as of now?
At the moment, it'd just be a Duke project. I mean, I would hope we can make it multi-institutional. I think it's really important to do multi-institutional studies because, that we need to make sure these things are scalable. So
I'm going to reference my computer for a second because I don't want to botch this.
Yeah, no.
You are currently the principal investigator of a novel immunogenic telomerase.
Yeah.
Mediated therapy in GBM project.
Yes.
Can you share more about that project, what the goals are, and what you're doing with that?
Yeah, sure. So this is a project we're doing in collaboration with UT Southwestern, and the principal investigator there is Professor Jerry Shea. So Jerry has been working on telomeres for his whole career. He, in fact, I don't think there's a person on planet Earth that knows more about telomeres than Jerry, probably.
For those who don't know, what is a telomere?
Yeah. So a telomere is...
I'm partially asking because I didn't pay a lot of attention in biology class in high school.
Well, so a telomere is the cap of DNA that fits on the end of your chromosome. And telomeres are involved in the aging process. So as we age, telomeres become shorter and shorter. And then once they get to a critical length, the cell can no longer replicate. There is an enzyme called telomerase. Okay, so there's telomeres and there's telomerase.
Mm-hmm.
So telomerase is the machinery that will extend telomeres. Now, in most cells in the body, telomerase is switched off, 0. And so that's why we all, unfortunately, get older. One of the reasons. There's other reasons as well. In cancer cells, telomerase is switched on. And when telomerase is switched on, the telomeres continue to stay the same length or get longer and longer, which means that the cell never dies. It never senesces. It's one of the hallmarks of cancer. That's interesting, and there's been lots of attempts to make drugs against telomerase, to shut telomerase down, some of which have been partially successful. What Dr. Shea discovered was that there is a particular drug that will take over the machinery of telomerase and insert rogue elements into the telomeres so that the telomeres shatter. By doing it's called 6-thiodeoxyguanosine. By doing this, what happens is the end of the telomeres become damaged and the immune system can see the cell, and then you get immune rejection of the cell. Why is that important in brain tumours? 85% of glioblastomas have what's called a TERT promoter mutation. TERT is telomerase. 85% of brain tumours, glioblastoma, I should say, hypothetically should be sensitive to this drug. And so we've done animal studies at our place at Duke, and we published this year before last to show that this drug is active in brain tumours in glioblastoma. And we're now working toward opening a clinical trial that would be available to 85% of glioblastoma patients based upon their TERT status.
That's amazing.
Yeah.
What does the trajectory look like for that?
So the Maya is the company. They're working with us. They have a study that's already open in non-small cell lung cancer internationally. They have had some good early results in that illness that also has this telomerase overactivity, and they're looking to fund our study. Probably, I think again it should open toward the end of this calendar year.
That's super exciting.
Yeah.
So a lot of exciting,
very exciting
clinical trials.
Yeah.
Very cool.
Yeah.
How does conceptualizing research, how does that work? Like obviously, incredible minds think of incredible things, but what does a process look like from, say, hey, I have this idea for maybe this drug that I've heard of, and then how does it actually translate into becoming a project?
Oh, that's a big question. When you're young, you generally look to other people and you work with them and identify what you think may be vulnerabilities, come up with hypotheses and test them. As your career unfolds, you draw on all your experience and others around you again to come up with hypotheses that you can test. And then you generally, as we discussed, test them in vitro and then in vivo and progress them if they look promising.
One of my last questions, you recently published an article about, the title was, Can Scientists Learn to Prevent Brain Tumors? For those that didn't read the article, can scientists learn to prevent brain tumors?
Yes.
Okay.
I think so. This is something I'm incredibly excited about.
Okay. Let's hear more.
Well, we think we can detect the cell of origin. So let me wind back 2 steps. One of the difficulties with brain tumours is at the moment, and I think this is in part probably true of many cancers, is that by the time we detect the cancer, it's already done a lot of mischief. So firstly, it's a large tumour by the time we detect it, and it's destroyed a lot of, brain, particularly glioblastoma, by the time we know it's there. So in order to cure that person or bring them back to good health, there's really going to have to be 2 parts. Firstly, we're going to have to cure the brain tumour with smart drugs and therapeutics. Second thing is we're going to have to get into regenerative medicine and regrow the brain or fix the damage that's been done. And, scientists are working away at that, but that's a fairly daunting task. If someone is paralysed or has lost their speech or their vision, how can you put that back together? Not impossible, but a big task. So, if we could prevent humans from getting brain tumours and prevent them from actually ever causing the damage that they do, that would be the holy grail of research. About a year ago, we came to a position where we think this is conceivable. With this cell of origin work, we think that, we know the cell of origin, we know where it lives in the brain, and then we've been using carbon dating-like techniques. You can use genomic time clocks to go backwards. So you and I, every cell in the body that we have collects, roughly speaking, 30 mutations per year. And you can use that information to date cells and see when a certain process started. By doing that, we think that brain tumors probably start very, very early in life, perhaps even in fetal development, the very first steps of a brain tumor. So if we could identify those cells at risk and we could see them in, for example, a 1-year-old or a 2-year-old, then if we could develop a therapeutic approach that could reverse that process or prevent that process, we could prevent brain tumors.
What would that look like, just like theoretical wise, where you're saying if, let's say, cells do start to formulate when someone's 1 year old, like how do you test that?
So
is that the question?
No, it's a great question. The very earliest events in the formation of a glioblastoma is duplication of chromosome number 7. We don't know why that happens, but it's an identifiable mark. So if you could identify either the reasons that that's happening in some people, or you could actually see those cells, you could image those cells in people before they accumulate the other events. So in order, roughly speaking, a glioblastoma has a duplication of 7, then it loses 10, chromosome 10, then it gets a TERT promoter mutation, then it gets a P53 mutation in many cases, and so on and so on. And it only becomes a glioblastoma when it's got a certain accumulation of genetic and other events. The cells that are the seed cells are not glioblastoma yet, and yet they have these early marks. So if you could somehow identify those cells with those early marks, perhaps you could give a drug or you could do something to prevent them from collecting those other events. Perhaps there's a particular environmental cause that people should avoid exposure to. Might be as simple as that. If I had a cell in my brain that had an extra chromosome 7, there's certain chemicals that I might need to avoid in the future to prevent me from accumulating other events and getting a glioblastoma.
You mentioned that when cells, gather enough of these different mutations, then it becomes a glioblastoma. But glioblastomas typically arise from grade 1, 2, 3, which is an astrocytoma typically, correct?
No, not a wild type glioblastoma. That in the setting of an IDH mutation, they usually go through grade 2, 3, and then 4. But in a glioblastoma, wild type glioblastoma, the first thing we know about them, they're a grade 4 tumor.
So then how would you say that if someone has like a grade 2 astrocytoma, are you essentially saying that ideally there would be some kind of like marker that would show that it would turn into a GBM? Because some people do live with astrocytomas and they don't progress into glioblastoma.
Oh, so we would hypothesize that every different brain tumor has a different type of cell of origin, and that the timetable and their aggressiveness is really in part determined by the cell of origin. So a grade 1 pilocytic astrocytoma in a child is probably coming from a particular cell of origin that is different from glioblastoma. A brain tumor, a medulloblasoma, which is in the back of the brain, will have a different cell of origin to a tumor that's in the front of the brain. And the nature of those tumors and the timetable and aggressiveness is in part determined by where they're coming from. So I would hypothesize that you can prevent all of them.
Yeah. That's fascinating. Like I just said, like I feel like talking to you for 30 minutes at this point, I've learned more than I have ever in any science class that I've ever taken.
Really? Well, that's very nice of you, but I'm not sure that's true.
I promise you it's very true. But it was a pleasure to chat with you.
Yeah,
Thanks
you
for
too.
coming on the show and sharing your knowledge. And I'm very excited for everyone to hear all of these incredible things happening. And I'm excited to see the progression of all these trials happening later this year.
Well, thanks for inviting me. It's a pleasure to be here.
Absolutely.
Thank you.
That's it for this week's show. Thank you so much for tuning in again to another episode of Glioblastoma, aka GBM. To get in touch with our organization, visit us online at gbmresearch.org or visit us on Instagram or Facebook at Glioblastoma Research. Visit us on Twitter at glioblastoma.org or visit us on LinkedIn at Glioblastoma Research Organization. To make a donation to the organization, which is fully tax deductible, visit us online at gbmresearch.org, where you can designate your donation in honor of someone or find other methods that you can make a donation with. Thank you again for supporting us, for supporting the show, and we'll see you next week. Welcome back to another Deep Dive with Staff Strong. Today, let's talk about this episode with Dr. David Ashley. What was your biggest takeaway?
Dr. Ashley, I thought it was really interesting how he talked about conducting studies and using mice as subjects, right? And oftentimes you'll see this, and he made really good points, right? It's cheaper, right? It's easier to genetically engineer mice and work with it when you're trying at a very early stage, whether it's a drug or looking at a specific mutation. But I thought it was fascinating and something I wasn't even fully aware of the difference of a mice brain to a human brain and how a lot of treatments aren't translating because of those differences, right? And hearing that as someone who funds research, and it'll be something I focus on way more as I look at it. But again, he talked, your point was Temodar, I think, right? And he said, yeah, that's one. Right? So many things that has great promise and they're really feeling strongly about after it's tested on a mouse's brain and they move it to a subject, it's falling flat. And I thought that was very interesting to hear from a leader in this space just say that plainly.
I thought it was interesting how when I first started the nonprofit, if you're not in the cancer space, right? Like you don't necessarily think about how they're conducting research. And so when I got into the nonprofit because of my father, I was thinking like, okay, so we're going to start funding research. And I started meeting with different institutions and talking about different projects. And then I was told that, one institution was doing mice trials. And I was like, how are you doing this, right? And then it kind of becomes like you understand, like you have to be able, it's like a weird, it's like an interesting ethical balance because on one hand, like it's an animal, right? But on the other hand, how are you ever supposed to test anything? So it's definitely this interesting, weird, gray area. I don't know
what's the most
how it works.
difficult part about brain cancer, right? Passing the blood-brain barrier. Right? So how would you ever emulate that? Right?
I had no idea.
Yeah. But so, I think hearing that is interesting as studies occur and as we look at funding, right? It's not always mice. Obviously, there's plenty of there's fellowship grants, discovery grants, plenty of other things that go on. But a very interesting viewpoint in the weeds, if you may, of research and like literally stage 0 research, well before anything goes to clinical trial drug development, as he was talking through that. And just like, again, if you missed this during the podcast, I'd invite you to go back and like listen to it again. I found myself rewinding 30 seconds at a time and hearing it again and writing different notes about that. And to me was something I learned at that point during his podcast with you. Yeah.
I also really liked how this episode was particularly very informative and extremely very descriptive, but in layman's terms.
Yep.
And even the tech that was helping produce that podcast, at the end of the episode, he was like, wait, like I learned a lot. And like, I've never heard any of these words before. Yeah. And so I think that episode or this episode will be very informative and helpful to a lot of people, not just on like how the back end of research works, but also just simple facts about brain cancer and how, like, why it's so complex. And I'm excited for everyone to kind of like listen to it and hopefully that makes a big impact too.
Yeah. And I think that's one of the most interesting parts to me about top institutions. You get a chance to sit down with someone like this and just hear him speak through what they're looking at, studies, and as you said, layman's terms. That's often, as I've seen and talked with oncologists, surgeons, researchers at these top institutions, that's how I find myself leaving feeling, because there's a reason they're a top institution in the country, right?
Right. Well, thanks for listening to another episode of Deep Dive with Staff Strong.